Welcome to another installment of the Library Seminar Series, Knauss Fellows 2020. I'm Lisa Clark, Reference Librarian at the NOAA Central Library. Today's library seminar features two 2020 Knauss Fellows, Michelle Harris and Tori Luu. And Christine Zinken, a 2020 Knauss Marine Policy Fellow with the Global Ocean Monitoring and Observing Program, will introduce our two speakers, each of whom will speak for 20 minutes, followed by a 5-minute Q&A. But before I turn this webinar over to our speakers, here are a few logistical tips to help you enjoy our presentation today. If you're having any trouble with your audio or visual components of GoToWebinar, please log out and rejoin us. This usually will resolve your technical issues. This presentation is also being recorded and will be available on the NOAA Central Library YouTube channel, as well as the Library Seminars website by later this afternoon. We'll also be accepting questions throughout the webinar, which the speakers will address at the end of each of their presentations. Please feel free to type your questions in the question box on the right side of your screen at any time during the presentation. I would suggest, however, since we have two presenters, that you distinguish to whom the question is directed, either Michelle or Tori, so that we know who your question's for. So with that, I thank you for attending this library seminar, and I turn the presentation over to you, Anne.
Thank you. Michelle Harris is a 2020 Knauss Fellow placed in the U.S. Integrated Ocean Observing System Program Office. Prior to the fellowship, she completed her master's degree in geography, oh my God, at the University of South Carolina, where she was a research associate with the Wind-Induced Nearshore Dynamics lab. Her research within coastal geomorphology have primarily focused on beach-dune interactions, coastal hazards, remote sensing, and GIS for management applications. And I'm handing it over to you, Michelle.
Awesome, thanks, Anne. All right, and thank you, Lisa and Anne, for having us here today. So again, my name is Michelle Harris, and I am a Knauss Fellow in the IOOS office, or the U.S. Integrated Ocean Observing System. I am representing the South Carolina Sea Grant today. And I am a student from the Wind Lab through the University of South Carolina in the Geography Department. And what we do in the Wind Lab is really focus on the beach and dune system, and more so looking at different metrics to evaluate impacts to our coastal communities, whether through field studies, remote sensing, or low-cost methodologies. And so today I come to you to give you a little bit more of a 30,000-foot presentation on some of the work that's come out of our lab this year, really focusing on understanding coastal dune response to tropical cyclones. Let's see here, there we go. So why do we study our environment here? And so we know that coastal populations are on the rise. We have increasing density on these coastlines. We also know that they're some of the most populated and economically important areas. As we have that continued growth on our coastlines, we also understand that that is coupled with our natural hazards. And so shown here, the central mechanism is a picture of a tropical cyclone. And we know recently now that these are increasing both in frequency and in magnitude. And so as we handle these natural hazards and protect our populations, we adopt multiple methods of coastal management. These take shape in either soft engineering or green engineering, such as the sand fences shown here on the right, living shorelines or beach nourishment, or more gray or hardened engineering methods such as seawalls, groins, revetments, and etc. The bottom line here is that our coastal dunes are the first line of defense against these hazards. So they serve really as a natural buffer or a sponge that absorbs either wave energy or wind impact that ends up preserving the landward infrastructure. So really important that we understand these systems and that we have them in place. So a little bit about the site that I will be talking about today. It is a barrier island off the coast of South Carolina. Isle of Palms, or IOP, is what we call a classic drumstick barrier island. You'll notice here that it has a thicker northeast end and it tapers off to the southwest. It's close to Charleston, approximately 16 or so kilometers off to the east-northeast, and it has longshore transport rates that taper from the northeast to the southwest. If you look here, you'll see that we have 3 sites labeled A, B, and C. 2 of which I will discuss in 2 different studies, and then Site C will be reserved for a 3rd study. So zooming in on those sites to see what they look like, on the far left-hand side here, you will see that we have Site A, or what we call our natural and control site. This is a beach dune system that's approximately 55 meters in width, and it's located on the northeast end of the island, close to a private community that has next to no foot traffic. This area is more of an incipient foredune zone that has been building over the last several years. Our Site B here in the center is what we call our anthropogenic site. This is located along Pavilion Drive on Isle of Palms, which is the main beach access point for visitors. And if you go landward of this site, you will see that you have a larger parking lot on the island. Lots of heavy foot traffic at this site here. You'll also notice if you look closely that we have trash cans from those people here along the coast. Now, if you look here on the far right-hand side, you'll see that it's broken off into sites C1 and C2. These are going to be reserved for the 3rd study that I will introduce, which is my thesis, where we looked at the implications of sand fences. So unlike sites A and B, which include both a subsection of the beach and the dune, Site C is reserved just for the dune system. Now, this is newer imagery, but on the left-hand side of Site C, that blue box did initially include 10 shore perpendicular sand fences, and Site C2 on the right-hand side was the control that had just a natural environment. So looking at Isle of Palms, I wanted to discuss some of the coastal management practices that are popular along the island. All 3 of these pictures are local to Isle of Palms and have been completed recently. And so if you look up in the top left-hand corner, that is an example of beach nourishment. That is completed where a ship goes offshore to a sediment site and dredges and pumps the sediment back onto the beach. This is done to widen the beach and is typically done after erosion that occurs on the northeast end of the island. And this has been completed back in 2010 and again in 2018. If you look at the center picture there, that is my thesis field site with the sand fences. And now these are installed typically either from homeowners or from city officials that are looking to build up your dune system for a low-cost metric. These sand fences are typically installed either shore perpendicular or shore parallel, and you'll see them on a variety of places on the island. I know I walked it in 2018 and there were approximately 21 installations. And the way that these work is really playing off of your aeolian transport. And so what happens is your wind will reach a certain threshold of shear velocity where it exceeds your threshold shear velocity. It'll carry wind-blown sediment longshore. And as that wind-blown sediment that's suspended encounters these fence slats, it creates a drag that can eventually lead to deposition. And so in this case, the sand will deposit on the lee side of the fence and slowly bury the fence and create a dune. In some instances, these are made of different materials, typically seen of these 4-foot wooden slats, in some cases also attached with vegetation to help stabilize that dune system. And then on the far right-hand side here, we see what is called beach scraping. On Isle of Palms, beaches are scraped typically post-storm under emergency orders, where sediment is taken from the foreshore and using heavy machinery is placed on the primary dune, pre-storm dune line. And so this creates what we call a mechanical dune, and that it's not a true natural dune. It can have a larger sediment grain size in some instances, and it's not typically vegetated. So they respond a little bit differently in some environments. For these 3 studies that I will discuss today, we are looking at these mechanical dunes over time that were placed post-Hurricane Matthew. So after we look at our sites, I wanted to talk a little bit about the different variables that we consider. First and foremost, this is one that I've grown a lot more accustomed to now with my time at IOOS and NOAA, but we rely on wind and wave data that we've used through NOAA NDBC or the National Data Buoy Center. Now from these data centers, we pulled multiple types of data, first and foremost being our wind speed, wind gust, and wind direction. And these are paramount when considering aeolian transport within a dune system. Now, similar to the fences, dunes will build with wind-blown sand that is carried longshore. And in a natural dune, it's typically trapped with different types of vegetation. And as it's trapped and deposited, it will slowly build up that dune system. And so we want to look for different metrics in terms of which way the wind is blowing and what speeds it's reaching if it's able to actually build up those dunes. We also pull our water data. So in this instance, we'll have wave heights and water levels, especially whenever we're considering whether or not a storm surge or king tide may have come in and impacted our site. Secondly, we look at precipitation. And for these studies, we have relied on COCORAS, which is the Community Collaborative Rain, Hail, and Snow Network. COCORAS has been around for roughly 20 years and is a citizen science initiative that we're really happy to partner with, where you can go online and pull either daily, monthly, or even annual precipitation, where local citizens have gone in and reported values from their rain gauges. There's also NCEI data, but for these three studies, we've primarily relied on COCORAS. And we look at precipitation for primarily understanding the impact to transport. If sediment becomes wet or inundated with precipitation, it is less likely to transport under wind conditions. And then lastly, we look at our actual environment or our site morphology, primarily looking at grain size. As we have coarser grain sizes, it becomes more difficult for them to transport along the beach. We also have been looking at vegetation density and type. I know one of my colleagues did her thesis at looking at dune builders and stabilizers and the impacts of different species, as well as the occasional prickly pear that we happen to walk upon as we're going through the dunes there, and then breaking it down to our field surveying, which I will discuss here in a moment. So I also wanted to remind you that Isle of Palms has been impacted by quite a few hurricanes over the last year, as has the whole East Coast. So the studies that I will mention today really pick up after Hurricane Matthew and start out at the post-Hurricane Matthew date for our surveying. I joined the Wind Lab in the fall of 2017, so I have been here and helping with field surveying and data processing through Irma, Florence, Michael, and Dorian. And I'm sad to say I've missed this hurricane season, but I know it's been an active one as well. And this is of growing importance, again, because we're recognizing that these storms are increasing both in magnitude and frequency. So again, taking this at a 30,000 foot level, I wanted to touch base on three different topics that have come out of our lab this year. The first being a post-storm dune recovery paper. So this is really the capstone piece that let me into the lab. I started it out just after Hurricane Matthew, and it really served as a teaching moment for me. It taught me how to do the field surveying, all of the data processing, and we created our own entire DEM processing step. So this is a multi-year longitudinal field study. We've been going out approximately every 5 to 6 weeks, and this study considers over 22 months of data. So for our field surveying, we go out with a team of 2 to 6 people typically with our Sokia total station, utilizing old rebar and bench posts for our benchmarks and backsite. We go out and take approximately 200 data points for topographic data that run from the secondary dune lines to the water line that run along shore perpendicular transects. Now, our data points are predominantly taken every meter apart or at geomorphic breaks, and we do our best to walk in straight lines as we can. You can see the data overlaid down there at the second image where I've taken several different survey dates to compare those lines. From those survey dots that you'll see there, I go ahead and process them and turn them into digital elevation models and separate the dune volume from the beach volume because we want to understand how the dunes themselves are impacted and have a more holistic view of the entire beach dune system and the sediment exchange that may occur there. The example image there, the third one to the left, is showing that process. So this is just an example generated in Google Earth where you see the dune highlighted in orange is separated from the beach or the upper foreshore in this example with a unique dune toe line. Now the dune toe lines are demarked for each survey date and are marked with our field surveying equipment and show a distinct change in slope between the dune and the upper foreshore. and they serve as the most seaward area extent of the dune. After we take those volumes and DEMs, we go ahead and create a digital or a change map, which you see on the far right-hand side here. This is showing the volumetric change over time, where the values in blue are showing rates of accretion and the values in red are showing rates of erosion. We further went ahead and overlaid our dune toe lines on these surveys to see if we had a seaward growth or a landward recession of our dunes over time. So that's just an example image there on the right. So here's an example of what this looks like looking at our data. So on the right-hand side, we're looking at an approximate year's worth of data where we looked post-Hurricane Irma all the way up to pre-Hurricane Florence. So from September of 2017 to September of 2018, and really mapped out not only the rate of recovery that we were seeing in volumetric values, but also where within our site we were seeing that erosion or accretion, so change over time. And in this instance, you're seeing the blue areas of accretion at the landward extent of the site, which are falling within our larger vegetation patches, but an overall loss in volume on the lower extents of the site or the low-lying areas. Off to the right-hand side is a figure I generated that really helps to sell this story and take it home, where you'll see polygons overlaid of the spatial extent of the dune. And so we have our landward extent that remains staple, as well as our extents on the sides, left and right, but our seaward extent is what is changing over time. And so our first survey date here, going chronologically, is showing post-Hurricane Matthew over time to pre-Irma, and then we saw a sharp decline in the dune width post-Irma. And so we had some wave energy come in that scarped the dune back to number 3 here, which really cut the dune's volume nearly in half, where it slowly started to recover over time. So some management implications from this study. Again, I wanted to hammer home that our longitudinal data sets are really sparse in the literature. Most researchers are able to go out and do pre-storm and post-storm surveys, but looking at this relationship over several years is pretty rare. So I'm really proud that our lab has been able to go out and sustain these studies. We also found that an alarming rate, our dunes really were not recovering between these storms. So over that 22-month data period, we saw that Site A, which was our natural site, still maintained an overall loss of 16% dune volume. And Site B, which was our anthropogenic site, lost nearly half of its volume and was severely scarped. This is really a concern for long-term sustainability, particularly with the rising frequency of storms that are hitting the East Coast. And so this is just an example image here of this walkover that did initially have dune coverage all the way to the edge of it that has been scarped and pushed landward. So the second paper that I wanted to touch on today is looking a little bit more in detail at the tropical cyclones as they come in. And so this is comparing 2 storms that impacted Isle of Palms, being Hurricanes Irma and Hurricanes Florence, that impacted the island as a tropical storm strength according to the Saffir-Simpson hurricane wind scale. And so we wanted to understand if 2 storms of similar intensities could lead to different geomorphic impacts on the island. And so very similar field methods here. We continue doing our field surveying, but for this study, we pulled in our pre and post hurricanes Irma and Florence DEMs. And so from those DEMs, I went ahead and generated the change maps, which are shown here as an example. And what I'll note here again is that your pre-storm, in this case Irma on the left, your pre-storm dune line is shown here in the dashed line, and your post-storm is shown in the solid line. So not only did we see that our dunes receded at this site, which is the anthropogenic site, but we see all of this red coloration noting that amount of erosion that occurred post-Irma. You'll also note here between these 2 values that you're seeing a greater rate of change for Irma here on the left than we're seeing for Florence on the right. So not only do the dune toe lines hardly migrate for Hurricane Florence, but your rates of accretion and erosion are drastically lower for Hurricane Florence. It's worthy of noting also that Florence came through South Carolina, whereas Irma never actually entered the state, but it had tropical storm force winds and energy near our site. So here's just an example and a quick overview of some of those storm characteristics. And so listed here are those values that we pulled from the Edisto buoy that's approximately 74 kilometers east-northeast from our site. If you look and compare at the energies here, you will see that Hurricane Irma, again, even without a direct impact, had a greater impact to our site, where we lost nearly 39% in volume compared to Florence that had a very minimal loss of 3% dune volume. I'd also like to note that Hurricane Irma was a very precipitation-driven storm, where our CoCoRa's local rain gauges were recording over 180 millimeters compared to Florence at roughly 32. And we were overall able to see that Irma really drove in a hydrodynamic influence. Our dunes were scarped, our dune toe was, kicked landward up to 20 meters, whereas Hurricane Florence, our dune toe hardly migrated, but we still saw some patterns of erosion and accretion within the secondary dune system, indicative of aeolian transport. And this is also possible by the lack of precipitation during Florence that could allow for that transport to occur with the higher wind speeds. So implications for this moving forward with management, we did note that even though both storms were categorized as tropical storms according to that scale, they did have drastically different geomorphic implications for our field site. And we have the dune volumes listed there, as well as the implications for how our sites are influenced, whether it is aeolian driven or whether it could be hydrodynamics. We went forward with this study and really highlighted how necessary in situ or site-specific observations could be. So we are relying on that offshore buoy, but given these storms come in from different directions and different intensities, we really want to adjust our methods in the future to have site-specific observations, whether that's through a MET station, anemometers, or just closer equipment that we're able to deploy and monitor these conditions. And then last but not least, just a brief mention of the third paper that is my thesis, looking at sand fences installed post-storm. On Isle of Palm, sand fences are traditionally installed after hurricane season, but after walking the beach, we noticed that in some cases they were left up throughout the hurricane season. And given they operate based on wind energy, I wanted to understand what relationship they could have if they survived through a hurricane season. So we adopted a different method here. This came out of a very late night conversation and wondering how we could get some new data for this fence site that we had found. This came out prior to Hurricane Florence and Michael, where we went down and deployed a measuring tape, PVC pole, and a hose clamp and ran shore perpendicular transects over Site C, which was that fenced and control site, for a variety of measurements shown here on the right using some handy field volunteers. From those measurements, I was able to generate some volumetric schematics, which I will spare you here, but if you have questions on them, I'd love to discuss them further. And we calculated the dune volume. We were able to look at macro scale changes in volume between different segments of the site, as well as look at micro scale using either rebar that we installed as erosion pin markers or the actual fence posts themselves as erosion pin markers. And so very quick method, we're able to get down there and survey the site within 20 to 30 minutes. We also don't have to rely on prior installed benchmarks or back sites for this method. So overall implications from that field study is that we not only created this method that can be rapidly deployed and gave us pretty accurate measurements of this volume, we also saw that the sand fences were beneficial during this hurricane season. Now, we did have erosion predominant during Florence for both the fenced and the control site, but we had a dune growth during Hurricane Michael within the fence site, which had higher wind speeds. So stepping out from this project, again, at a 30,000 foot level, we really wanted to highlight how this could be an adoptable method that either coastal communities or citizen scientists could use to go out and take quick measurements or to serve as an educational resource. We also took this project and tried to predict aeolian transport by grain class and modeled that out based on the conditions that we were measuring with that offshore buoy for wind and with the different size sediment grains that we were sampling at the site and sieving within the lab. And so something else that came out of that, as well as the implication for HEMS, or what we call human erosion machines. One limitation here is that beachgoers do like to remove stuff that they find, and so some of the erosion pins did grow legs and wander away. So some quick wrap up with ongoing research, challenges, and strengths. First and foremost, some challenges here. Site accessibility, being 2.5 hours from the coast, does have its limitations. You can see here on the right, we were, I think, the second car in contraflow getting off the island during Florence, trying to quickly get our measurements. Site-specific observations, as I said, we're hoping in the future to really have our own MET station or some other methods for monitoring our conditions. Fieldwork pests, I don't have pictures here, but mosquitoes, fire ants, cacti, oh my, always fun, as well as rain and little pop-up storms that, as you can see here, can be difficult for our equipment that we have to protect. And then strengths, ongoing collaborative effort. I couldn't be here speaking about these projects without my lab mates, so I'm really happy that I can discuss these with you guys today. And this is an ongoing research need as we face more and more incoming storms, as we increase our density along the coast. This is something that will need to be studied. So quick acknowledgements. Again, these are my teammates. My advisor, Dr. Jean Ellis, leads this lab. Dr. Mayra Roman Rivera was really my teacher. She was the senior PhD student when I came on. And then the rest of my co-authors and lab mates, again, I couldn't present this work without their help as well. And with that, thank you. I'll be glad to take any questions.
Thank you, Michelle. I appreciate your presentation. We do have one question, but before I read it to you, I just want to encourage everyone to add questions. We're going to take the next 5 minutes to do a Q&A, but if we don't have enough time to get to your question, we will send it to Michelle. So this first question asks, regarding the difference between impacts from Florence versus Irma, did you consider the wind direction as a factor to dune erosion, onshore versus offshore?
Sorry, mute button. Yes, we did look at wind direction, and Irma came in from the northeast and kind of traveled obliquely longshore, whereas Florence came in from the east-northeast and traveled in. So that was one thing that we considered, especially with Florence being more of an aeolian impact and looking at how the sediment was transported within the lower lying areas of the dunes. Irma wasn't as significant within some of the areas of the dune sites because we really saw such a distinct change from the scarping, and practically half the dune system was eroded and pushed offshore after Irma. But wind direction is definitely something that we considered, and one thing that is a challenge moving forward because that buoy is located offshore, and we would love to have, a site-specific anemometer to really capture those changes.
Excellent. We have another question. Any use for drone-based imaging in studying geomorphology changes?
Yes. I'm really excited, and I hope to say that we can move forward and use drones. I know right now we've been using the lab-based equipment, which has been the total station and prisms. We've looked at an RTK unit, and I am hoping to utilize drones in the future. I know that's something we're investigating.
Anyway, I don't see any more questions. I'll give it a second to give people an opportunity to type them up in our questions chat box. Meanwhile, I'm going to change the screen and give it to our next speaker, Tori Luu.
All right. Thanks, Lisa.
Thank you.
And I'll be quickly introducing Tori. Tori Luu is a 2020 Knauss Fellow in NOAA's Office of International Affairs, supporting policy work in the Arctic, Cuba, Sargasso Sea, and other international fora. She is finishing up her PhD in ocean biochemistry at Princeton University using isotope geochemistry to study nutrient cycling in coral reefs. Her research has focused on ground truthing the use of coral skeletal nitrogen isotope ratios as a proxy to understanding environmental nutrient dynamics. This work has taken her from the low nutrient waters of Bermuda up to the upwelling zone of the equatorial Pacific.
All right, hello. Are you able to see my screen?
Your screen is blank right now. Is that
Interesting.
intentional? There you go.
It's
Oh,
funny
there
because
we are.
we just tested this.
Yeah. I see your screen now, but it's not on PowerPoint mode.
On presenter mode.
Mm-hmm. There you go.
Okay, hopefully that holds. Okay. Hi all, and thanks again for spending your lunchtime with us today. My name is Tori Luu, and I am a graduate student at Princeton University working in partnership with the Bermuda Institute of Ocean Sciences. I am a Knauss Fellow this year in NOAA's Office of International Affairs, and I am representing the New Jersey Sea Grant Consortium today. So today I'll talk to you about the portion of my PhD work that I've carried out in Bermuda. And this talk title characterizes my work that's framed around evaluating how we can use the nitrogen isotopes of organic matter in coral skeletons as a proxy for studying changes in the nitrogen cycle. The question here really is, how well do corals capture the changes going on in their environment when it comes to recording what they've been eating? So stony corals date back to the Triassic period, about 240 million years or so ago, and they became the important reef builders that we know today about 25 million years ago. And because corals lay down skeletal layers over time, coral skeleton cores have been used to generate high-resolution records of temperature, pH, pollution history, and much more, which is really helpful when learning about the environment when we don't have direct measurements for that time. So common geochemical proxies that have been used in coral skeletons are focused on the inorganic component, which is mostly what you see here. Carbon and oxygen isotopes or elemental ratios such as strontium and calcium are relatively easy to measure at these high resolutions. But what about the nitrogen isotopes that I'm talking to you about today? This is relatively new. Nitrogen is in the organic matrix that corals lay down as a blueprint during calcification, but it's less than 0.03 weight% of the total skeletal material, which makes it tricky to isolate and measure. This was especially difficult previously because when it was explored in the early 2000s, the traditional methods required at least 1 micromole of nitrogen or about 500 milligrams of skeletal material, which made high resolution records basically impossible. And I won't go into the details, but some newer methods of measurement can now reliably measure tens to hundreds of nanomoles, which is 1 to 2 orders of magnitude less material. The method that I use requires only 10 milligrams of skeletal material, which allows us to start generating these higher resolution records. This is super exciting because it allows us to get a better look at nitrogen cycle over time, which is highly dynamic and has important implications for primary productivity and thus the rest of the marine food web. Many of you are probably familiar with the marine nitrogen cycle shown here. There's a lot going on, but some of the key pieces that we're interested in is how inputs and outputs to the pool of fixed or biologically available nitrogen changes over the course of Earth history. So generating high-resolution records with coral skeletons can provide insight into some historical anthropogenic changes. These processes can be distinguished by looking at nitrogen isotopes. So as a quick sort of primer, there are 2 stable isotopes of nitrogen, nitrogen 14 and nitrogen 15, which has 1 more neutron and is slightly heavier than nitrogen 14. And many biological processes have some degree of bias towards the lighter N14 because the chemical bonds are energetically easier to manipulate. So you can think of this as, for example, a kid who has a preference for regular M&Ms over peanut M&Ms because they're easier to chew. And so they're not going to say no to peanut M&Ms, but as they're picking out the M&Ms out of the jar, there might be a slight bias towards the regular M&Ms. And after the kid has eaten a chunk of the M&Ms from the jar, the ratio of peanut M&Ms to regular M&Ms is going to be a little bit higher than what you started with because the kid preferentially ate more regular M&Ms. A different kid might have a stronger preference or less of a preference for regular M&Ms, and this is what I mean by biological processes having these different degrees of fractionation. So in terms of isotopes, we track this through something called the delta notation, so delta N15 shown here, which is the ratio of 15N to 14N of the sample, so like the leftover jar of M&Ms, for example, compared to the N15 to N14 ratio of some standard. For nitrogen, the standard is N2 gas in the atmosphere. And the unit of this ratio is per mil, or per 1,000, instead of percent, which is per 100, because these preferences are pretty slight. So in this very busy diagram, we show how different biological processes impacting nitrogen in the marine environment, for example, denitrification or N2 fixation, have different isotopic signatures. It's not really important for you to know all of the ins and outs of this right now. But for example, water column denitrification tends to elevate the delta N15 of remaining nitrate in the water column, and it happens in low oxygen conditions. So delta N15 records can help interpret changes in oxygen level over time, for example. Anthropogenic nitrogen inputs also have specific isotopic signatures. So delta N15 records in coral can be used to track human-driven nutrient input, as another example. Looking at a map of mean nitrate delta N15 measurements that we have for the upper ocean, we see that nitrogen stable isotopes can act as a natural tracer for various nitrogen cycle processes. However, also looking at this map, you can see just how sparse the measurements are, and that's just in the modern ocean. So we really do need a tool if we want to study how things have changed in the past, and that's where the corals come in. But given how this proxy is still relatively new, it's important to thoroughly evaluate how robust of a tool coral skeleton-bound delta N15 really is. This next figure shows that the coral skeletal delta N15, which is shown on the y-axis, seems to roughly track the delta N15 of nitrate that's supplied to the surface ocean, shown on the x-axis. When you're looking at the broad global scale, these points are from all over the world. This is really promising for being able to use coral delta N15 records as proxies to represent the delta N15 of nitrate supplied to the surface, and thus you could treat coral delta N15 as reflective of environmental nitrogen cycling processes. But it's not always perfectly clean, and there are still some uncertainties. One of these is the biological effects that might happen separate from what's going on in the environment. So from that, the question arises of how well does a coral record actually capture the signature of the nitrogen that's available to it in the environment? This is a big question because corals have photosynthetic symbionts, or zooxanthellae, which can acquire nitrogen in many different forms and recycle nitrogen internally that's sort of separate from what's going on in the environment. So a 2015 study measured coral skeleton delta N15 and zooplankton delta N15 as the presumed food source or primary source of nitrogen to the corals. And here we see that coral delta N15 in the solid bars over these sites increases moving inshore to Tynes Bay about 3 per mil, while the plankton delta N15 shown in the white bars moving inshore is only increasing about 1 per mil. So this is an example where the delta N15 recorded by the corals doesn't seem to match the environmental patterns. And the question then is, how can we possibly explain this? Looking at differences in conditions across those sites, we see that the estimate for food availability showed that there's about 2.5 times as much food in Tynes Bay inshore than offshore in Hog Reef. And turbidity also tends to be higher in Tynes Bay than in Hog Reef, which could affect light availability. The reason that this is important is because there are a couple of different internal cycling hypotheses for how corals can cycle nitrogen internally as waste products from the food. So take, for example, this case of efficient cycling, where the coral is just really good at not leaking any of its waste. The coral will eat food and metabolize it like we all do and produce some amount of nitrogen waste because it has these endosymbiotic algae. The algae can uptake the nitrogen waste products and keep it within the coral system rather than losing it to waste. In this case, the coral delta N15 should look exactly like the food because you are what you eat. It's not losing anything from it, at least nothing that is fractionating. In the case of inefficient cycling as a hypothesis, say for example, the coral is eating a lot of food, and it doesn't need to be quite as efficient in the internal recycling of nitrogen waste products. So it'll leak some of this waste out, and if that biological process has a slight bias or preference for the lighter isotope, that'll change the delta N15 of the coral relative to the food. And if there is sort of a gradient between these two, there's a range, and these processes differ under different feeding conditions, that can really have a negative impact on the coral's ability to record changes that are solely based on the isotopic composition of the environment that they're in. I looked at this problem in two different ways. One was to conduct a laboratory feeding experiment that's highly controlled, and the other was to go out into the environment and redo that study that I showed you earlier, basically, and try to capture it in a little bit more robust way. Starting with the feeding experiment, we tested the conditions of feeding rates and light levels by crossing two feeding rates, high fed and low fed, with high light and low light conditions. The high light conditions were not quite as high light as you would get on the reef, but still different enough. And the feeding had a fourfold difference between high fed and low fed. We collected juvenile corals on these tiles shown in the top right to make sure that in the experiment we're getting all new tissue. So you can see here growth on the tile, the same tile over time. And we reared them in a highly controlled environment with light and temperature controlled, and even used this elaborate algae filter to strip all the seawater of dissolved inorganic nitrogen to make sure that the only source of nitrogen would be the food that we fed them. Taking a look at some of the experiment results, we see that the high-fed corals grew faster than the low-fed corals, with high-fed corals about tripling in size by the end of the experiment, where low-fed corals barely doubled in size. But light did not seem to have much an effect here. Taking a look at the nitrogen isotopes, we measured the total coral, which is shown in the yellow points, but also we separated coral tissue and coral symbionts to help us better understand internal nitrogen cycling. For our purposes, we can focus on the yellow points of the low-fed and high-fed corals compared to the food. So this is delta N15, and we see that the high-fed corals and the food look basically the same. And even for the low-fed corals, they're not that different from the food. There's less than a 1 per mil difference here for a fourfold feeding difference. And if you'll recall from that previous experiment that I highlighted, the corals that had 2.5 times more food available were more than 2 per mil higher than the food. And so this is a pretty good indication that perhaps the internal cycling processes based on feeding availability is not as dramatic an effect as one might have thought. We use all these data to reform the model, but we won't go over that today. Really, I think that the main takeaway for this question is that under a fourfold difference in feeding regimes, coral delta N15 really didn't change that much, which is good for us and good for the proxy. But since the original hypothesis no longer explains that 2015 data that we saw, our work isn't really done here. And so just highlighting the conclusion from the feeding experiment, where coral delta N15 seems to depend mostly on the nitrogen source with little influence from internal changes due to the different feeding levels or light levels. We try to address the question by going back into the environment and doing this field study. So the original study looked at corals and compared it to zooplankton in the water. And the difference there is that corals are fixed at the bottom, and they're always going to be recording what's going on in that specific location. The zooplankton are moving in and out with currents and with their diel patterns, and so what you capture in one grab of water might not necessarily be representative of the average that the coral sees over time. So in this field study, we compare corals to other benthic organisms to test their ability to capture broad patterns. Revisiting that same figure that showed how in Bermuda the coral delta 15 appears to increase more than the food source. The question is, are we measuring the food source accurately? And what would we get when we do look at these benthic autotrophs and heterotrophs? So the reason that we selected those macroalgae for autotrophs and filter feeding Christmas tree worms for heterotrophs is because they're also fixed in the same position and represent the ends of the spectrum that coral might fall between in terms of their ability to acquire nitrogen. And so the macroalgae and the Christmas tree worms, they don't have that same complex internal nitrogen cycling with symbionts that corals do, and so they should be able to just straightforwardly capture changes going on in the environment. If we compare corals to these organisms, we can see how they do comparatively. And the reason that we still would like to use corals over these presumably more perfect organisms is because the macroalgae and the worms don't really leave behind fossil records for us to examine the way that corals do. So we want our corals to match these other guys. Looking at the delta 15 gradients across the Bermuda pedestal, here is Hog Reef offshore and moving inward towards Tynes Bay inshore of the island. So it's just a map view of those sites that you've been seeing in the red, orange, green, and blue. And we looked at 3 different species of coral, macroalgae, those serpulid, Christmas tree worms, and also we did a grab of the particulate organic matter and zooplankton as potential food sources. So moving to some of the results, if you measure the delta N15 of all of those components as we did, and then plot them in a map, what you see is that from offshore to inshore across those points that I just showed you, the patterns are pretty much the same across the board, even though the absolute values are not. But the corals shown here in black, ability to capture this pattern of change of increasing delta N15 follows pretty closely to what you see from the macroalgae, the phytoplankton, the zooplankton, and also the serpulid worms. And another way of looking at those data is to compare the coral delta N15 directly with the macroalgae delta N15 and the serpulid worm delta N15. And so when you plot coral delta N15 against the macroalgae, you see that these points fall mostly on a line with a slope close to 1. So having that 1-to-1 relationship with a slight offset. Similarly for the serpulid worms. Excuse me for 1 second. But what we're saying here is that corals living in this environment do a pretty good job at capturing changes in the nutrient conditions compared to the other benthic organisms living in the same environment. And namely those that don't have a potentially complex internal nitrogen cycling. Taking a closer look at the different species of corals that we examined, we do want to note that those measurements I showed you before were about the coral tissue itself and not the skeleton material. So how does the coral tissue relate to the skeletal material? The tissue delta N15 shown here is plotted against the skeletal delta N15. And again, you see these slopes of 1. And so there's this 1-to-1 relationship between the coral tissue and what's recorded in the skeleton. So that should mean that the patterns recorded in the skeleton are reflecting what is in the coral tissue and therefore sort of a timestamp of what's going on in the environment. I do, however, want to note that there are differences between the coral species. And so while the slope is 1-to-1, the absolute value and the offset varies from species to species. And there are these species-specific offsets. So this places some limitations on our ability to produce records when you have to combine multiple species of fossil corals, for example, when you do like a big grab and you don't really know what species are there. You might not be able to glean as much from that. However, records generated from a single species of coral should be quite robust because of these relationships. So we return back to this sort of roadmap of how we address the problem, and we found that in this grading experiment, the coral delta 15 does in fact capture environmental patterns of nitrogen sources about as well as other benthic heterotrophs and autotrophs. And from earlier in our feeding experiment, we did learn that coral delta 15 depends mostly on the nitrogen source. And putting those things all together, I think we can conclude that if you have a good handle on the uncertainties with coral skeleton-bound delta 15 as a proxy, then corals really can provide a robust, high-resolution record that we can use as a tool to study any sort of changes in the nitrogen cycle. In essence, corals are what they eat, and we can take advantage of that for understanding environmental change. And with that, I just want to throw out some acknowledgements there to my advisors from the Bermuda Institute of Ocean Sciences, from Princeton, and from IOOS. A lot of the technical staff and interns at IOOS who helped with the field collections and laboratory setup, a bunch of undergrads at Princeton who helped with measurements, whose senior thesis projects I advised, and a graduate student in particular who trained me in all these methods early on and whose figure I continue to bash and question and break down in this talk. But thank you all for listening, and I'll take any questions that you have.
Fantastic. Thank you, Tori. We do have a couple of questions, which we're going to take the next few minutes to answer. And just as a reminder to the audience, if you do have questions, please don't be shy about asking them. Just type them into the question box on the right. This first question asks, do you have any sense for how long organic matter is preserved in coral skeletal tissue? In other words, how far back in time could you reconstruct environmental variables from nitrogen isotopes in the organic material?
Thanks. Yeah, that's a really good question. And so part of the history of using nitrogen isotopes to look at and records through time is that originally people were just taking cores through the sediment and measuring like bulk delta N15 of those parcels of sediment coming through the water. And there were a lot of questions about how well the nitrogen is still preserved, what nitrogen you're actually preserving, how much of it has degraded. The reason that our lab moved towards these mineral-bound measurements is that the nitrogen is theoretically preserved pretty well, and you can look for diagenesis signals, but because the nitrogen is like part of the blueprint itself that the calcium carbonate is built around, that calcium carbonate, if still intact, should be preserving that nitrogen material. And so we've gone back several million years, and my lab mate using foraminifera, which also produces calcium carbonate, has recently produced records which are some of the oldest records going back to like 65 million years. And so there's a lot of potential. You can also look at these really old fossil corals and measure the delta 15 changes and try to identify when symbiosis started, because that would be a different isotopic signature.
Excellent. Our next question asks, how would you expect this to translate to mollusks?
Sorry, could you repeat that?
Sure. It asks, how would you expect this to translate to mollusks?
To mollusks. Yeah, that's also a great question. Mollusks, I think, are a little bit trickier because they, I would say that they live more in the sediments than corals do. And there are a lot of different nitrogen processes that happen in the sediments that are a little bit isolated from what's happening in the water column. And so I think people have tried to measure delta 15 in mollusk shells and get environmental signals from it, but there is this different imprint because they're like digging and eating up in the sediments instead of feeding from the water column, or those two signals are mixed. And so I think it's harder to interpret environmental signals from mollusk shells. But there are a number of like mounted mollusks that might have potential as well. The methods would be very similar.
Excellent. Well, we do have more questions, but our time has run out. So do you have any last comments, or Michelle or Anne?
Just thank you for having us with you on this webinar today, and thank you for organizing.
Agreed. Round of thanks for everybody.
No, we appreciate having you guys here. So one last reminder to the audience, if you joined us late or you have a colleague you think might enjoy this Knauss Fellows webinar, please send them the recording. You can find it on the NOAA Central Library YouTube channel or the Library Seminars web page later this afternoon. I'd like to conclude by thanking our speakers for sharing their research with us, and to Anne for introducing Michelle and Tori today. And again, to the audience, we truly appreciate that you joined us for today's library seminar. The library is proud to present the work of the NOAA community, to support the Knauss Fellows, who are awesome, and we hope you will join us again. So be well, everyone. Thank you.